Welcome everyone. I Dr. Shamishta Banerjee welcome you all on the SWAM course on genetic engineering and its recent advancements. Today we are going to deal with future trends in transgenic animals. So today's video lecture is the fascinating world of transgenic animals and the future trends that are poised to redefine biotechnology and medicine. Over the next 30 minutes we will delve into the cutting edge development in animal genetics and how they are reshaping agriculture, medicine, scientific research and many other fields. Even in our daily life examples, we can have a overview of it. So, here are the quick subtopics, quick glimpse of the subtopics which we will be dealing today. That is, firstly, in short, I will be taking you through introduction to transgenic animals, current applications of transgenic animals, future trends and innovations, ethical and regulatory considerations, as well as conclusion or the key takeaway points or call to action, because this is such a topic which is the need of the hour, and in sooner or later there will be many transgenic products into the market. So, it is the need of the hour to have what is the call to action, what is our take. So, that also we are going to discuss in today's lecture, that is basically opinion about the ethical considerations. Okay. Till now in this subject we have studied much more things like transgenic animals, I will focus on it, as well as we have also studied the process of transgenesis and how the transgenic plants as well as microbes are created, that also we have discussed. But in further lectures again we will discuss what is the future trends involved in this transgenic animals, or what are the applications that are into the recent trends or that are into the market, we can say. Let us begin by establishing a foundational understanding of transgenic animals. In detail we have already studied. From the name itself, by now we can say what are transgenic animals? They are organisms whose genetic material has been deliberately altered by introducing genes from some other species. It is used to integrate the exogenous gene into the animal genome by genetic engineering technology, so that these genes can be expressed and inherited by the offspring. Why we are calling it as exogenous gene? Because it is a foreign gene, it is not present in the host organism, that is why it is coming from outside. We are having the gene of interest in the form of our exogenous gene. Okay. So, this process typically involves identifying a specific gene, isolating it and then inserting it into the animal's genome, often with the help of many advanced techniques like CRISPR-Cas9. This also we are going to study in detail about this technology, the mechanism involved in CRISPR-Cas9 as well as the case study of CRISPR-Cas9 or genome editing techniques, we can say, in further lectures. So, before going into the future trends, the historical context is again very important. So, let us have a review of all the historical landmarks which have taken place in case of transgenic animals. Transgenic animals have been pivotal in advancing our understanding of genetics and have opened doors to exciting possibilities. The groundwork for transgenic animals was laid in the year 1960s. See how early it was laid, when scientists began experimenting with transferring the foreign genetic material into the genome of animals. So, in 1970s the first successful gene transfer was done or was performed, we can say. This breakthrough paved the way for more complex genetic manipulation in the future. In 1980s, mice and the first transgenic animals were created. 1980 saw a significant leap in transgenic animal research. In 1982, scientists successfully created the first transgenic mice by introducing the foreign DNA into the embryos. In 1985, researchers isolated and cloned green fluorescent protein, which is abbreviated as GFP gene, from a bioluminescent jellyfish. The discovery revolutionized the genetic research, allowing the scientists to tag and track specific proteins within the living organisms. This was a case of reporter gene, we can say. The gene which does not report its presence is basically tagged with this GFP protein, and as a result, if we want to know the expression of this particular protein, we can phenotypically mark the presence or expression of such gene which is not having the reporter activity. So, this was a landmark discovery in the field of genetic research, we can say. During 1990s, transgenic technology expanded to a wide range of animals including rats, rabbits, sheep and pigs. Transgenic animals were created for various purposes such as pharmaceutical protein production as well as even for agricultural improvements. In 1996, Dolly the sheep, the first mammal, was cloned from an adult somatic cells, which marked a groundbreaking achievement in transgenic animal research. Although Dolly was not a transgenic animal herself, her creation demonstrated the potential for cloning and genetic modifications. So, it is included under the historical achievements under transgenic animal segment itself. In late 1990s, custom made transgenic animals were created. Towards the end of 1990s, the technology for creating custom made transgenic animals improved significantly. The 21st century has witnessed a proliferation of transgenic animal research with diverse applications. Transgenic animals have been used to study human diseases, produce pharmaceuticals, enhance agricultural productivity and even explore regenerative medicines, or we can say the reparative medicine. The current applications of transgenic animals we are going to discuss in further slides. Let us explore some of the present day applications of transgenic animals. Transgenesis allows improvement of nutrients in animal products, including their quantity, the quality of the whole food and specific nutritional composition. Transgenic technology could provide a means for transferring or increasing nutritional beneficial traits. For example, enhancing omega 3 fatty acids in fish consumed by humans may contribute to a decreased occurrence of coronary heart diseases. In fact, transgenic pigs that contain elevated levels of omega 3 fatty acids have been produced. Furthermore, transfer of a transgene that elevates the level of omega 3 fatty acids into pigs may enhance the nutritional quality of pork. So, these were the examples where the transgenic animals were created for enhancing the nutritional composition of any particular livestock, we can say. Recent progress has produced prion free and suppressed prion livestock. Now, what are these prions? Prions are basically the causative agents. These are smaller than the virus particles. They do not have any cell envelope or cell coat, and these are responsible for causing infection. So, a very well known prion which is known till date to cause infection is bovine spongiferum, that causes mad cow disease or BSE, that is bovine spongiferum encephalopathy in cattle, and in CJD in humans. What is this CJD? It is Creutzfeldt-Jakob disease in humans. So, these are the diseases which are mainly caused by prions. So, it is the need or the requirement that the livestock must be created which could not be infected with these prions. Transgenic animals are used to study fundamental biological processes, like for example, they can help elucidate the role of specific genes in development and physiology. Transgenic animals are crucial in studying human diseases, drug development and testing several novel or new therapeutic modalities. For example, mice can be genetically modified to mimic human conditions like cancer, Alzheimer's disease as well as diabetes. So, these are the diseases which are more prevalent in the society, we know. Hence, to study the mechanism of disease or to study the treatment modalities or for the discovery of the different drug approaches, the animal models needs to be created. So, with the help of this transgenics, we can use them as a model organism. These models will help the researcher understand the disease mechanism and test the potential treatment method. It can also be an application in drug development and testing. Transgenic animals are used to evaluate the safety and efficacy of new drugs. They can be engineered to express specific drug targets or exhibit even the disease symptoms. That allows researchers to test the potential therapies. That means, by using this transgenic animals, we can know whether a drug is effective or not, or we can know whether a drug is capable of reducing the symptoms or reducing the disease in that particular organism. So, for this, again this is a very landmark application of transgenic animals. It can also be helpful for biopharmaceutical production. Biopharmaceutical production, pharmaceutical means these are the proteins or these are the products which are of pharmaceutical importance or which are of therapeutic importance. Biopharmaceutical means they will be created with the help of livestock, with the help of living organisms. Normally, pharmaceuticals can be produced by other chemical approaches also. So, here that is why we have specified them that transgenic animals are used for creation of biopharmaceuticals. Animals like goats and rabbit are used to produce valuable proteins and antibodies for medical purposes. Some transgenic animals, particularly goats, rabbits and cows, are engineered to produce therapeutic proteins in their milk or other bodily fluids. These proteins are used to develop medications for various diseases. Transgenic animals can also be used to produce vaccines. For example, chickens have been genetically modified to produce vaccine components in their eggs. Agricultural applications. In agriculture, transgenic animals can be engineered for improved meat and milk production as well as for creation of disease resistance organism. Transgenic animals can also be engineered for improved agricultural outcomes. For example, cows can be modified to produce milk with specific traits, or livestock can be made more resistant to diseases. Conservation efforts, like we are hearing since a decade that many organisms are on the verge of extinction or mainly have already become the extinct species, or some are categorized under the tag of endangered species. So, the conservation efforts can also be done with the help of transgenic animal creation. Like, transgenic animals are playing a role in conservation by helping restoring the endangered species. For instance, researchers are exploring ways to use transgenic techniques to help restore the endangered or extinct species. Future trends and innovation. Now, let us delve into the exciting future trends that are on the horizon for transgenic animals. Till now, we have discussed how the transgenic animals are created for these applications. Now, further we will discuss what are the products that are in pipeline or what is the future scope for the creation of this transgenic animals. Disease resistance, some have already been created. In the future, we can expect some animals which are genetically modified to be made more resistant to diseases, thus in turn benefiting both agricultural as well as human health. The most important application of transgenic technology is manipulation of MHC. What are these MHCs? They are major histocompatibility complex. They are basically a set of genes which influence the immune response and increase the disease capacity of livestock. Clements et al. In 1994 reported that the transgenic sheep have been developed that is resistance to Visna virus infection. The transmission of BSE, that is also known as scrapie disease, is also prevented by the knockdown of prion proteins. The knockdown technology has already been discussed in details in previous lecture. Transgenic mice have been developed for the secretion of recombinant antibodies in milk to neutralize the coronavirus, which is responsible for transmissible gastroenteritis, that is TGEV, which is an economically important disease in case of pigs. Transgenic dairy cows are also being created that secretes listostaphin into their milk and have higher resistance to mastitis due to the protection provided by listostaphin, which kills the bacteria or the causative bacteria, we can say Staphylococcus aureus, in a dose dependent manner that protects the mammary gland against the major mastitis causing pathogen. Biomedical advancements. Transgenic animals are poised to revolutionize organ transplantation, or we can say xenotransplantation, reducing the availability or the waiting window for the donor organism, with the potential for animals as organ donors for humans. Environmental adaptation. Climate change may prompt the development of animals engineered to thrive in changing environments. Like there are animals which could survive the extreme cold condition, we can say polar bear, penguins, and that means they have that particular type of gene so that they can survive. So, we can produce such livestock which can even survive in that extreme condition. Or we know that the livestock more popularly known as like Jersey cows. These are a breed of cows which comes from the north region. Nowadays, inbreeding is basically done to have a hybrid with the cows which can sustain the all environmental condition, but having the quality of milk just as that of Jersey cow. So, these are the techniques by which environmental adaptation barrier can be overcome by creating such transgenics. The human health. The main potential application of transgenic animal is the production of recombinant and biologically active proteins in the mammary gland, and this in turn could be used for the benefit of mankind. This is also called as gene farming. Mammary gland is the preferred site for the production of these proteins because large quantities can be extracted as well as purified. Moreover, milk is a secreted body fluid that is normally produced in large quantities and which could be collected without causing any harm or pain to the animal. Transgenic animals can also serve as a valuable model for studying and developing treatments for human diseases. It can also be helpful for studying the disease model, already I told you. So, in past also it has been attempted, but in future what could be the exact example of this? That is, farm animals like cattle and pigs could be used as an appropriate model for the study of human diseases. Till now we have been using mouse or guinea pig as the human disease model, but this is the future trend. Like diseases like cystic fibrosis, cancer and several neurodegenerative diseases like Parkinson's, Alzheimer's and their therapeutic approaches can also be studied by using cattle and pigs as transgenic disease models. Pigs could be also used as an effective model for the study of growth hormone releasing hormone defects. that is GHRH defect. Genetic editing advancement is also an application of future trends. Advancement in genetic editing technique will enable more precise and efficient modification in animals. Environmental monitoring. Some transgenic animals are being explored as environmental indicators. For example, fishes are engineered which changes the color in presence of pollutants. They can also serve as early warning systems for water quality issues. So, by this the different environmental pollutants can be measured, or we can say the water quality indexes can also be assessed by creating transgenics which can monitor the environmental changes, monitor the minute environmental changes we can say. Organ transplants, already I told you that the waiting period for organ donors, or we can say xenotransplantation. Like what is xenotransplantation? From some other species, using the organ of some other species, it is also known as interspecies transplantation, or in short it is known as xenotransplantation. So, transgenic pigs are being developed as potential sources of organs for human transplantation. Researchers are working to genetically modify these pigs to reduce the risk of organ rejection. That is, during the process of xenotransplantation, this is the main challenge we can say, that is not accepted by the host organism. There are many issues, and as a result allergic reactions or immune responses is elicited or heightened, and because of that immunosuppressive drugs are also provided, and again the compromised immune system is observed. So, these are the limitations, hence by using the transgenics these limitations can be reduced we can say. The extraordinary success of human to human, an example of intra-species, or you can say allotransplantation. Xenotransplantation is basically interspecies. Intra means within the same species, but the organisms are different. So, allotransplantation was successfully obtained in case of vascularized organs like heart, kidney, liver, lungs and pancreas, and it has saved many lives over the past 25 years. But it has also created a significant need for donor organs. It was recognized early on that for physical, anatomical, ethical and supply reasons, the pig was the best choice as a donor animal for vascularized organs. Vascularized organs means which are vasculative in nature, that means which have a hollow structure and through which the blood or any other fluid or the connective tissues can move or float we can say, they are basically known as vascularized organs. However, serious immunological issues had to be overcome before the pig to human transplantation model could become a reality. These transgenics can also be used for regenerative medicine, already I told you. More specifically, it can be used for repairing the damaged tissues using induced pluripotent stem cells. Now, what are stem cells? I will just give you a glimpse of it. Stem cells are those cells which are undifferentiated and they can proliferate throughout their lifespan or divide throughout their lifespan. So, undifferentiated means they are not specialized, or we can say their functions are not specific till now. So, these are normal stem cells. Now, based on the potency level, the stem cells can be categorized into many. Coming to pluripotent stem cells are a bit specialized cells we can say. So, these pluripotent stem cells can give rise to a cell of the same lineage, like hematopoietic stem cell is an example of pluripotent stem cell. So, pluripotent stem cells are advantageous in comparison to the unipotent stem cells. That is why in this case for tissue repairing technology, the pluripotency is basically induced in case of unipotent cells we can say. So, using induced pluripotent cells, that is iPS cells, were directly injected into the vitreous of the damaged retina of mice. there is the layer which is present in the eye, that is vitreous layer. So, this iPS cells were directly injected into that layer. The stem cells engraved into the retina growed and repaired the vascular vessels in the eye of that particular transgenic mice. This is a case for tissue repair in regenerative medicine segment. Ethical and regulatory consideration. While the future of transgenic animal is promising, it raises important ethical and regulatory questions. Ethical concerns include animal welfare, environmental impacts, and the potential for unintended consequences that cannot be monitored or that cannot be measured, we can say. Hence, a robust regulatory framework is essential to ensure the safe and responsible use of transgenic animals, or we can say the GRAS segment, that is which have been termed as genetically recognized as safe animals or safe organisms. Specifically, we can say it can be transgenic plants as well as transgenic animals depending on the circumstances. The social opinion on transgenic animal research is divided almost in the middle. Like in different segments of the world, different countries of the world, the social opinion varies. Opinion survey in USA, Japan and New Zealand revealed that only 42, 54 and 58% respectively of the people participating in the survey favors such researches. That means, they accept such creation of transgenic animals or use of such transgenic animals. The main reason for opposition of people were as follows: use of animals in biotechnological research causes great suffering to the animals. This is the belief. Actually, this is not the case always. So, it is felt that by using animals for the production of pharmaceutical protein will reduce them to mere factories. Some people feel that animals should be regarded as equals to humans, in that they have the same basic rights as human beings or same basic feelings as human beings. But an argument is attempted to focus on the integrity of such species, in that each biological species has a right to exist as a separate identifiable entity. But biologists do not regard a species as a fixed watertight entity, rather they are regarded as dynamic, constantly evolving groups. Finally, the introduction of human genes into animals and vice versa may be seen by many as clouding the definition of humanness. That means, our integrity is getting distorted. Many people think in that way. That is why again its acceptability is not that worldwide. But most of the known human genes, they are not unique, and comparable genes do occur in animals. In addition, many retroviruses have integrated into the human genome without any recognizable devaluation of our humanness. Limitations of transgenics. The transgenic technology, even though has tremendous application in livestock improvement programs, still it has lots of limitations. Insertional mutation resulting in alteration of important biological processes may occur. Insertional mutation means in between, actually the transgene is not getting at the desired site, and that is why some mutation can generate, that is basically known as insertional mutations. Unregulated gene expression resulting in improper expression of gene products. Possibility of side effects in transgenic animals like arthritis, dermatitis and even cancer. These are the major limitations. Conclusion. In conclusion, the future of transgenic animals is filled with potential to transform medicine, agriculture and conservation. In the mid 1980s, the advent of transgenic technologies generated great excitement in the scientific community and the pharmaceutical industry. So, in today's class, we have studied what are the future trends of future prospects of these transgenic animals, how they are created and how they are used in our day to day life. Here are the key takeaways of this lecture. Transgenic animals are at the forefront of scientific innovation. Future trends include disease resistance, biomedical advancements and environmental adaptations. Ethical considerations and robust regulation are very much essential for the responsible and safe use of these transgenic animals. So, call to action thing is again very important. As a society, we have a role in shaping the future of transgenic animals through thoughtful discourse, responsible research and ethical engagement. So, thank you for joining us on this journey through the world of transgenic animals, and in the next lecture we are going to study the farm animals. Farm animals are basically the animals which hold a pharmaceutical importance, or we can say the animals which are created through farming. It is PHARMING, do not confuse it with farming, that is FARMING. So, gene farming is basically a segment where the genes are expressed on the milk or any livestock kind of thing, or in egg we can say. So, the therapeutic proteins present in these transgenic animals are known as farm animals. So, together we can unlock the new horizon in biotechnology and medicine. Hoping to join us in the further lectures. Thank you for joining today. Thank you.